This video will cover part 2 of subtopic 2.5, enzymes. So, a list of understandings, applications, and skills that will be covered in this video. And this will cover the use of enzymes within industry. So, enzymes are widely used within industry, and they are used as immobilized enzymes. So, immobilized enzymes are enzymes that are attached to a material so that their movement is restricted. There are several ways in which this can be done. We can get aggregations of enzymes bonded together, so you cluster a bunch of enzymes together to create that aggregate. Commonly, they are going to be attached to a glass surface, or another common way in which they get immobilized is through gels. So, you can have these gel beads, which are made from a compound called alginate, and the enzymes will be adhered to the surface of those beads. So there are advantages of using immobilized enzymes. The enzymes are easily separated from the product, so it's easy to get the product and leave the enzyme behind. The enzymes are easily recovered and can be used again for whatever process is needed. And immobilizing enzymes will also help increase their stability, so you can get a lot more use out of them. And the advantage of immobilized enzymes is that they can be exposed to higher enzyme concentrations than usual. So immobilized enzymes have many common uses in various industries. So for example, enzymes get used in detergents or found in detergents. So for example, proteases and lipases are often common ingredients in detergents and will be used to break down proteins and fats. Production of biofuels will make use of enzymes, so things like amylase will be used to break down the starch in grains. They can get used in the process of brewing through clarification of those products. And enzymes can be used in medicine and bioengineering. They can be used in diagnostic tests to test for the presence of something like HIV, for example. They can be used to clean contact lenses as another example. So one way in which enzymes get used in industry is through the production of lactose-free milk. So lactose, if you don't remember, is a sugar that's naturally found in milk when lactose is broken down. So lactose is a disaccharide. And when it gets broken down or hydrolyzed, it gets broken down to glucose and galactose. And this gets done by the enzyme lactase. So that enzyme lactase will take lactose and split that disaccharide into its individual monosaccharides of galactose and glucose. So there are several advantages of producing lactose-free milk. So some people are lactose intolerant. It's a common condition in adulthood that adults no longer produce lactose. So, people who don't produce lactose cannot consume dairy products. Glucose and galactose are actually sweeter, so less sugar is needed for milk products, such as yogurt. And cheese production can be faster. So bacteria ferment glucose and galactose a lot faster. So if we remove the lactose or break down that lactose for the bacteria, it removes a step that they have to do. And then in production of ice cream, the presence of lactose can actually cause crystallization, creating those crunchy ice crystals. So removing that lactose can help improve the shelf life of ice cream. So there are some different methods of producing lactose-free milk. One of the methods, which obviously relates to this lesson, is through enzyme mobilization. So it is possible to make the process of generating lactose-free milk more efficient by immobilizing the lactase on a recoverable surface such as alginate or alginate beads. So first the lactase gets immobilized on alginate beads, so the enzyme gets coated on the surface of those beads. Next, the beads are placed in a container over which the milk can be passed. So the diagram illustrates this process here. So the yellow circles represent those alginate beads. Those alginate beads have been coated with lactase. So on the surface of those beads is where you find that immobilized enzyme. Milk is then poured over those beads containing the lactase. The milk can then be collected as it passes over those beads. So as the milk passes over those beads, the lactase that is present will break down the lactose. That milk can then be recirculated to ensure that all lactose is converted to glucose and galactose. So this model of an industrial process allows the lactase to also be recovered and reused, which makes the process cheap. We get an efficient conversion of lactose to glucose and galactose, and reduced purification of milk, since the enzyme is retained and a higher percent of lactose conversion is achieved. So there's less steps in purifying the milk at this point. All these factors work together to reduce the cost, particularly on the downstream processing and purification of the milk after the milk has been passed over the lactase. So one of the other aspects that go with using enzymes is being able to design experiments to test the effect of things like temperature, pH, and substrate concentration on the activity of enzymes. As discussed in the previous video, these are all factors that can affect enzyme activity. So when designing an experiment, you have to consider what possible independent variable would affect the enzyme activity. So as mentioned in the title, things like temperature, pH, substrate concentration are all possible independent variables. Another possible independent variable would be the presence of an inhibitor. So these are all things that can be manipulated, that could then be used to study the effect on enzyme activity. So once you have a possible independent variable identified, you need to identify possible dependent variables. And these would have to allow enzyme activity to be measured. So possible ways in which enzyme activity could be measured, and this would really depend on the enzyme that's being used. Could be things like gas production, for example. So the gas production can be sensed or detected by gas sensors or by water displacement. Other possible dependent variables include things like digestion of a solid, where a percent change in mass could be measured, or volume of liquid produced could be measured. Or digestion of a liquid, where diffusion out of a dialysis tube is measured, or color change as well. So there's various tests that can be done. So for example, the iodine test for the presence of starch, the presence of peptide bonds using a compound called the Burette test, or the presence of sugars using a different compound called Benedict's reagent, can all allow for enzyme activity to be measured indirectly. So the following chart just contains examples of enzymes, substrates, and products. So we have catalase, which decomposes hydrogen peroxide into oxygen gas and water. There's the enzyme diastase, which acts on starch and converts that starch into maltose. There's pectinase, which acts on pectin, which is found in plant cell walls, and will produce simple sugars or break down that pectin into simple sugars. And then there are the proteinases, pepsin and renin, where pepsin will act on protein to break it into short polypeptides, and renin will take soluble casein, which is a milk protein, and convert that into essentially curdled milk. So the renin will essentially curdle the milk. So all examples of enzymes and substrates, and then how you would measure enzyme activity would vary on the type of product that is produced. So for example, catalase, because oxygen gas is being produced, this could be measured through water displacements, for example. With the pepsin, for example, because the substrate is protein and that's breaking that down into short polypeptides, you could use the Burette reagent or the Burette test to test for the presence of peptide bonds. So that is the end of this video for part 2 of subtopic 2.5 for enzymes.